Okay, today we'll be discussing epidemiology, which is tracking infectious disease. Okay, looking at epidemiology, we're going to first get into some of the vocabulary to define the types of outbreaks and diseases that we see. Following that, we're going to look how epidemiologists actually track the movement of disease within a population. And we're going to look at how technology and climate has changed the transmission mode of disease throughout the centuries. And at the end, we're going to talk about the emergence of new pathogens and what we can do about it. Okay. Looking at the chain of infection, epidemiologists are going to study a wide range of aspects of transmission of disease in order to fully understand and take a holistic approach on what is exactly going on. So first of all, we can start by identifying the organism of interest. We'll see how we can identify the organism of interest by testing of the organism itself. We want to also not only know the identity of the organism, but the virulence of the organism. So, how infectious is this organism? How dangerous is it to humans? And how invasive is it? Following that, we want to also look at the overlap of the reservoir. Is this organism held in the forest? Is it in ticks, on deers in the forest? How do we get close to this reservoir? How do we deplete this reservoir? How easily then is it transmitted to humans? Is it going from animals to humans and then from human to human? Is it airborne? Is it vector-borne? Can we get it from touching a doorknob like a fomite? And then we have to look at the host itself. Looking at the host, we can understand the chain of infection, not only of how the disease gets to the human, but how the infection actually enters into the body. So that would be the portal of entry that would be of interest. And looking at the hosts themselves, we want to look at their immune system. So how effective is their immune system? Are they very young or are they very old and have a compromised immune system? Are they pregnant? Are they immunocompromised from one way or another? This could alter the susceptibility of the host and the transmission of disease throughout the population. Let's go ahead and go back to John Snow, who's known as the father of epidemiology. He was one of the first scientists to track disease. In London in 1854, he was able to find the source of a cholera outbreak. So cholera was being transmitted throughout London, but people weren't necessarily sure how this disease was moving through the population. So what he did is he actually started looking at maps of the city and started mapping out people who were infected, where they were. What he found was there's a commonality between the infection and where people went to get their water. So everybody going to the same well was getting cholera. So he indicated that this well has to do with something with the transmission of disease. So they ended up closing down the well. People had to go to other sources to get their water. And what they found out is that the levels of cholera started dropping within the populations. It was being transmitted throughout the water in the well, that specific well. So by tracking down disease, he was able to break the chain of infection by closing down this well itself. Okay. Looking at epidemiology, we have descriptive epidemiology and analytical epidemiology. Starting with descriptive epidemiology, this is person, place, and time. We want to look at how many new cases are coming about. Are we seeing all of a sudden an outbreak of disease? On the other hand, analytical epidemiology is more about surveillance and looking at forming a hypothesis. So these can be observational studies or case-controlled studies of looking how disease is potentially spreading through a population and which risk factors are involved. Okay, some terminology to start with here. We have endemic, epidemic, and pandemic. When we look at these, we actually see an escalation of disease. Disease is tracked over time. So here on the x-axis, we see time, and on the y-axis, we see number of cases of disease. We're typically looking at a geographical location to start with, and we look at endemics. So we can see endemic over time, these small peaks, and then we see a larger peak, and that's going to be our epidemic that we're seeing there. And that would be in a regional area over a set period of time. When we track disease, we go endemic, epidemic to pandemic. An endemic is kind of a steady, low rate of infection. We typically find these from animal reservoirs. When we're living close to a forest, there's an animal reservoir harboring the disease, and people are going into that forest. Maybe hunters are going into that forest. We're going to see a low rate of exposure. And so we are not necessarily surprised about this low rate of infection. Epidemic, on the other hand, is something that happens over a short period of time, and we see an increase of infection within a population. So just like this graph here, endemic to epidemic, epidemic is what puts something on the radar for epidemiologists. Moving on to pandemic, this has to do with the worldwide spread of the disease. So we could be looking at diseases like COVID-19, which we're quite familiar with, HIV, qualified as a pandemic, it's worldwide, and influenza as well, sweeps across globally. So endemic to epidemic, then to pandemic. Again, endemic is low rate. Epidemic is that short period of time where we get a rise in cases. And pandemic is when it spreads worldwide. Okay, the term outbreak here is when we're looking at endemic levels of in a disease, and an outbreak is when we start seeing this spike that takes us to the epidemic level here. So from endemic to epidemic is where we see these outbreaks, and this is really when things go on the radar for the CDC or for any epidemiologists. This is when we start looking into disease reservoirs. Have this changed? Do we have people going closer into nature where these disease reservoirs are? We try to find out how this disease transmission spiked. So we look at all of the available variables that we have in this case of why something has gone from endemic to an outbreak leading to an epidemic. Okay, and what we try to stop at this point is this moving to a pandemic. So observing endemic to epidemic, we would see an outbreak, and we want to ensure that outbreak does not go global. Diseases don't normally stop at any kind of boundaries. There's no borders that they stop at. They're airborne. People are jumping on planes. So things can move very, very quickly from epidemic to pandemic. Okay, let's go ahead and discuss emerging disease versus re-emerging disease. So an emerging disease is one we find that recently appeared in the population. So a new disease, this is potentially a new variant of a disease. We discussed before that viruses can recombine in one cell, and this leads to novel viruses. So that's where we could get an emerging disease, something we've never seen before. On the other hand, re-emerging disease is a known disease where we all of a sudden see it start peaking, where it's coming up more than the endemic level, and we are watching out to see if there's some kind of outbreak, moving it to epidemic levels. When we see these increases, again, we have to look at the environment around us. Here we see an example of deforestation. Deforestation means people are getting closer and closer to potential reservoirs within forests. So as we move into the forest and we are removing trees, we're getting closer and closer to new viruses, new bacteria, or even re-emerging disease. Something that was in low numbers in the population can come out in higher numbers due to an increased amount of the exposure. Let's go ahead and talk about prevalence versus incidence. These are two important terms to know. Looking at prevalence itself, we're looking at the total number of active cases in a given population. When we're looking at incidence number, these are only the new cases in that given population. So prevalence is total number, that accumulation over time. Incidence is essentially the new cases popping up. So these are two descriptive terms that can look at emerging disease or even at emerging disease or known disease as well. How do we get from prevalence to incidence? Let's go ahead and look over this example here. So it's this example at Happy Valley Rest Home, 10 new cases of pneumococcal pneumonia occurred in March. For the entire month of March, a total of 23 cases were recorded. HVRH had 200 residents that month. That's Happy Valley Rest Home, HVRH. So let's go ahead and do some math here. We have incidence rate and prevalence rate. Incidence, again, is the number of new cases, and the prevalence rate, on the other hand, is going to be the total number of active cases. We know that there's 200 people, 200 residents at HVRH, the Happy Valley Rest Home. So let's go ahead and start with the math here for pneumococcal pneumonia at HVRH in March. So here we see incidence rate, the new cases. We had 10 new cases of pneumococcal pneumonia out of a total of 200 residents. So that's 10 divided by 200 times 100. That gives us a 5% incidence rate. Let's go ahead and look at prevalence rate. On the other hand, we have pneumococcal pneumonia at the Happy Valley Rest Home. We have 23 total cases were recorded. So that's going to be our prevalence, that 23 out of 200. So 23 divided by 200 times 100 gives us a prevalence rate of 11.5%. Now, if we're listening to the news and we hear somebody talk about incidence rate versus prevalence rate, a 5% versus 11.5% can sound very, very different. So because prevalence is the accumulation over time and incidence is just what's occurring currently, these new active cases that we find within the population, this can really convey a different message. So if a broadcaster wants to really have more of an impact, they would probably say prevalence rate over incidence rate because that percent is higher. Our prevalence is 11.5 versus incident rate is 5%. We also want to make sure that we're comparing apples to apples. If we hear one day we're at 11.5%, then the next day we're at 5%, we have to realize we're looking at prevalence rate to incidence rate there, and that these are different ways of analyzing disease. Looking at incidence statistics, we can see a couple different chronological views of measles, mumps, and pertussis, which is whooping cough over time. So while some of these are slowly being eradicated, others are peaking. So it's interesting to watch the differences in these. And again, these are incident rates. When we talk about incident rates, here we're looking at the number of new cases. So that can be graphed over time. Otherwise, if we were looking at prevalence, the graph would just continually increase over time because that rate only goes up. Prevalence, on the other hand, can go up and down, as we see here. Okay, let's talk about HAIs. These are called hospital-acquired infections. These are really important for allied health students. Hospital-acquired infections can be tracked in a couple different ways. One of the first things we always want to do is identify the isolate. So, are all of the patients that we're seeing with the current outbreak, are we seeing that they have the same exact strain of the microorganism, the same variant? So is this actually what's going person to person in a hospital setting? We also want to see which hospital staff members have made contact with the patients, because this point of contact could mean the spread. We also want to look at medical instrumentation. So what kind of fomite transmission could be taking place? Is it on incubation tubes? Is it in food, water? What is being passed from person to person that may be spreading the disease as well? We also want to see how the patients are in contact with one another. In this hospital, are patients remaining in their room, or are they coming out of their room and walking around and communicating and touching things? Do they have shared bathrooms? This is going to be really important to understand whether if this is person-to-person transmission and what kind of things that the hospital staff and the patients are coming into contact with. So things in a hospital setting that are important are going to be hand sanitization. So we have washing hands or hand sanitizers. Hand sanitizers work quite well. This should be done in between rooms as a method for controlling the spread of disease. There's also disposable garments. So this is part of the PPE, the protective equipment that we wear, depending on what the hospital setting is. PPE can be changed between rooms, so that's changing gloves between patients or changing your gown between patients. That way we're not spreading disease around. We know that MRSA, or MRSA, which is methicillin-resistant Staphylococcus aureus, has notoriously been passed around the hospital. So again, changing that PPE, washing hands is critical to prevent the spread of disease. Aseptic technique is really important as well. When we're looking at the techniques used in the hospital setting here, we see we're directly connecting to someone's vascular system, and we can accidentally inject a microbe into someone's vascular system, contaminating them to the point where they could end up with an infection and potentially spread it to others. So aseptic technique is really important. What we see in this image is called a daisy chain. So we're going to go ahead and analyze the daisy chain or the spread of disease in this hospital setting. We're going to be looking at nursing homes and hospitals, as we know patients move from nursing homes to hospitals quite frequently. Let's go ahead and look over this, and then we will discuss. So here we see the source is hospital number 1. We have 19 cases in 56 months. So here we're looking at years span, only 19, multiple years span. Then we move down to patient number 1 being transferred to a nursing home. And we have 14 cases now in 22 months. So we've now seen an increase in the number of cases here in less time. So that is a concern. Following that, we have patients from this nursing home moving into other nursing homes or into hospitals. So we see patient 2 is transferred to nursing home 2, patient 3 is transferred to the hospital, and patient 4 is transferred to hospital 3. Now, if these 14 cases are any of these people being transferred, we quickly see the spread of this disease from hospital 1 to a nursing home to infiltrating additional hospitals and nursing homes. Here we see hospital 4. We're seeing 12 cases in 32 months, and then hospital 5, long-term and acute care hospital, 11 cases in 3 months. So if we start out this 19 cases in 56 months over the course of years here, all the way down to 11 cases in 3 months. So we can see how quickly the disease can spread between hospitals and nursing homes. And this is the daisy chain, the chain of infection of what these small movements of patients with disease can cause in these hospital-like settings. And this is why infection control methods are so important. Okay. Let's talk about patient 0, or what can also be known as the index case. This is the first known person with the disease. When it came to HIV, there was a lot of stress on finding patient 0 and the patient who brought HIV into being a pandemic and who spread it globally. Initially, they had traced it down to a flight steward who they thought had transmitted the disease globally. This flight steward was actually named and vilified for this case, and they found out they were actually wrong in the end, that patient 0 was not the correct patient 0 for HIV. Now we have better techniques with epidemiology to track down our index cases and understand the chain of infection. The point of this is not to put blame on someone or to advertise patient 0. It's to track the disease. If we understand the profile of the disease, we can slow the transmission or stop the transmission. So here we see patient 1. All of these blue dots are the patient that they've came into direct contact with around them and have transferred the disease to. Then we can see moving from there, this person coming into contact with this person, or this person coming into contact with 3 more patients, and this one coming into 3 more. And we see this branching out quite a bit. So we move from 1 to 6 to 35 to 130 to 127, and we just see the movement of disease from patient 0 into the population. Okay. When we talk about surveillance of disease, we have some barriers up, and that's that we don't have modern reporting mechanisms in all countries. So we have the CDC and the WHO who work on reporting data and reporting data for global health awareness, but not all countries have the availability to do this. So the WHO sends out teams of scientists to these areas where they're less likely to have modern clinical laboratories. They're unable to effectively have case reporting within the population due to maybe resources or lack of staff, trained staff. And we also see even political systems can change, and it can affect the way that we can detect and limit outbreaks of infectious disease. So CDC and WHO help mobilize teams to areas so that we can have a global surveillance of the passing of disease. And this is how we can pick up on things that we know are endemic to epidemic, and that's when we get a spike for an epidemiologist to start looking into outbreaks and preventing them from becoming pandemics. Okay, we have a case study here, and I think we should read over this case study. So this is in Wuhan, and we're looking at the beginning of COVID. I like this case study because as you are moving through your life, and COVID affected us one way or another, I want us to see kind of the direct impact of where it started on the people there, and I want us to um, look into empathy and understanding of these situations and know that this could happen anywhere worldwide. Okay, so we have Peng Yang was a 29-year-old respiratory and clinical illness doctor at First People's Hospital in Jiayang district of the city of Wuhan, China. Near the end of December 2019, Peng found himself heading to work, daydreaming about his upcoming wedding, a long-awaited ceremony that would take place in just a matter of days. Before he couldn't, before he couldn't know what was about to happen. Later that morning, the patients started arriving. There were only a few at first, but a few grew to nearly 20 that day. Each of them had a high fever and a persistent cough and struggled to breathe. Radiographs showed that most had bilateral lung infiltrates characteristic of pneumonia. So, again, getting into kind of the day and place of Peng's life, this 29-year-old respiratory doctor, he's seeing a lot of people coming in with signs of pneumonia. So many people coming in with signs of pneumonia. Thinking this was just a normal day, it piqued his interest because that's not what they typically saw. The next day, more patients arrived, and even more the day after that. What was happening? Peng found himself on the front lines of what would be called COVID-19 pandemic, caused by a new emerging coronavirus, eventually called SARS-CoV-2. Peng knew he would have to postpone his wedding because he was needed there. Sadly, despite taking medical precautions, Peng himself was infected while treating his patients. By January 25th, 2020, nearly 700 patients with COVID-19 disease had been admitted to the hospital of Wuhan. One of those admitted was Peng. 5 days later, this selfless doctor died from respiratory failure. His wedding invitations, never mailed, were still in his desk. So I think this story, this case history here of Peng is a realistic view of where we go about our daily lives. And for people working, interested in working in the medical field, seeing how something like a pandemic could emerge from day to day, and where we think about the redundancy in hospital settings, that all of a sudden pandemics really break through and change. And then here we also see, again, the selflessness of the medical staff working within the hospitals who have this high risk job, high risk of exposure, as we see here. So again, COVID-19 has affected us in all different ways. I think this personalized story is something of interest because Peng died without knowing that a pandemic even started. So he passed away in 2020 without knowing the outcome of this. Okay. Looking at global pandemics, so SARS-CoV-2, we're going to be looking at some numbers here. So we're going to look at the movement of time. Back in October of 2020, so we had a well-established pandemic by then. We see 32 million cases, 1 million deaths. May 2022, we see 527 million cases, 6.28 million deaths. And from the WHO's reported data, because data reporting changed from that time, we see about 778 million reported cases and 7.1 million reported deaths. We know that SARS-CoV-2 is still circulating worldwide, and it is seasonal as well. While there's surveillance on SARS-CoV-2, it's not surveilled in the exact same ways that it was initially, and that's why we may see numbers changing from one reporting station to another. One of the main ways that we monitor disease in a population is actually looking at wastewater contaminants. So we can take waste, which contains fecal matter, and within fecal matter, we're able to pick up microorganisms and viral DNA or RNA, genetic material that we can see within the population. Is it increasing this viral genetic material or decreasing in the population? So we can actually test populations like this without individually testing a person. Wastewater can be tested. Of course, we have hospitalization data too when patients come in and get diagnosed, genomic testing. But it is a little bit different looking at how we report COVID today versus before. And I think this is a great image here. This is a transmission electron microscopy image, and we actually see SARS-CoV-2 in this colorized image in blue. So we see it right next to the cell. So that is the virus that has claimed so many lives, and we're able to observe it here. Okay. Looking at COVID-19 as a pandemic, we can start looking at the R number here. So this is on the x-axis, days after 100 confirmed cases, and cases on a log scale. So we can see how different countries were affected by the pandemic itself. The R number here is the number of potentially infected by one infected individual. So one person, how many people did they end up affecting? So this has a lot to do with our surroundings, whether we're using proper quarantine protocol, whether we are washing our hands, disinfecting the surfaces around us. The rate of transmission can change. The R number can change. So depending on the country, we see that the R number changes. Okay. Looking at how technology has altered the way disease spreads, travel is one of the biggest things. So, going back to even the Black Plague and the transmission of the Black Plague, that was actually transferred through travel routes from Asia to Western Europe because of the Silk Road. So it was able to travel. We also have merchant ships which were spreading disease. But today, the transmission of disease from one continent to another can happen in less than 24 hours. Someone can jump on a plane from one continent and be on another continent in, let's say, 16 hours or so. So we can have this global transmission really quickly. So what started as an endemic and then maybe hit an outbreak to epidemic levels, in less than 24 hours, the pandemic can already be emerging. And places like airports are going to be these grounds where not only is one person bringing it to another continent, but they're in contact with other individuals that are then transporting it into other areas. So these environments that we're in, where transport is taking place, again, it's not only moving from one continent to the next, it's also moving into other people who are taking it to other continents. So air travel has really changed the transmission of disease, and accessibility to air travel has as well. So being in these airports, being in international terminals with other people is an instant way that not only within 24 hours are we taking it from one continent to the next, we could be taking it to multiple continents within that 24-hour time span. Okay. Not only is travel affected at blood banks, so blood banks test for disease transmission to ensure that when people are getting a blood transfusion, that they don't have blood that is contaminated with HIV or hepatitis. So today we monitor blood. We know that also the loss of habitat and species diversity has caused diseases to get closer to the human population. So as we move into forested areas, we have closer exposure to these environments. So the suburban sprawls that we're seeing, suburban areas moving into the forest, is just bringing us closer and closer to disease. If we look at technology influencing emerging disease, again, these are new diseases that come out. So like we looked at the swine flu, that was a combination of avian and human viruses. These are novel viruses. These can be crossed continents within 24 hours. So on this map, what we're seeing here is emerging and re-emerging infections that epidemiologists, like ones that work at the WHO or the CDC, are tracking. So this is what's continuously going on: surveillance of the areas, monitoring prevalence, monitoring incidence rate to try to prevent the next global pandemic. Okay. Looking at re-emerging pathogens, so we've seen some changes. When we have an increase of immunocompromised individuals, whether that be from lupus, other autoimmune diseases, something like HIV, or we have a lot of young individuals and older individuals, any kind of immunocompromised person makes our population more susceptible to disease. So when we saw the global spread of HIV and AIDS, we actually increased the number of immunocompromised persons worldwide. So this was really concerning because when we have more immunocompromised people, we have more people susceptible to infection. which then can trigger more outbreaks because as the immune system is lowered or decreased and less effective, we have more infected individuals and more people being able to spread disease, and that can lead to more pandemics. So looking at immunocompromised individuals, we have some of them that were susceptible to Mycobacterium tuberculosis. So we see tuberculosis coming back out as a reemerging disease due to more people being immunocompromised at this point. Again, with the treatment that we have today, these retroviral drugs, people are not as immunocompromised as they were before with an HIV infection because it prevents the virus from replicating. So these medications are extremely important for keeping people healthy and the people around them healthy as well, and preventing re-emerging diseases. So we're starting to see a turnaround in this as well. Okay. Let's go ahead and move to the question section of this lecture. Question 1. Washing hands frequently will interrupt the link in the chain of infection. Okay, let's go ahead and you can pause it, read through the answers, and we'll move on. The answer to question number 1 is transmission of the pathogen. So, washing hands frequently will interrupt the link, which is that transmission of the pathogen in the chain of infection. Okay. Question number 2, a study in which individuals are randomly assigned to receive a vaccine or a placebo and are followed to monitor side effects and effectiveness of the vaccine is an example of what type of study? We'll let you read through the answers here. We have experimental, case control, observational, and cohort. Can pause here and we'll go ahead and pick up with the answer for question 2, and that would be an experimental study. So if we look back at the question here, we're using a randomly assigned people that are going to be placebo versus experimental with the vaccine. We've divided this into 2 categories, and we want to monitor the effectiveness of this vaccine. So we're comparing the control group versus the experimental group. So this is what we would say is an experimental study. Question number 3. The question says, during the month of May, 2 residents of an assisted living facility with 100 residents developed new cases of sepsis caused by multi-drug resistance, KPC. Molecular surveillance identified an additional 8 residents with KPC. What is the prevalence of KPC infection in this population? We'll give you a second to pause the video here and go ahead and do the math. Okay, so again, we're looking at prevalence here. We can go back to our slide with the mathematical equations, so they're not too difficult here. Here we go. Prevalence rate. So here we're looking at the number of patients over the total number of patients. So this is total number of active cases. We're not just looking at new cases, active cases. So we take total number of active divided by total number of patients times 100 to get that percent. So let's go ahead and go back to this question now. And we see we have 2 residents in a facility with 100 residents here. But then we have 8 additional residents. So what we do here is the 8 plus 2 gives us 10. And we're dividing that by 100 and then timesing it by 100. So 2 plus 8 is going to be 10 divided by 100, and then times 100 is going to give us that 10%. Okay, question number 4. Diseases that are always present in a community, usually at low, more or less constant frequency, are classified as having a blank pattern. Go ahead and pause here to address the answers. We have endemic, pandemic, epidemic, and prevalence. Moving on to the answer for question 4, diseases that are always present in a community, usually at a low, more or less constant frequency, are classified as an endemic pattern. Yeah, and that's endemic because, again, moving from endemic means that we're seeing maybe there's a reservoir around, seeing people with cases of Lyme disease getting closer to forested areas. We're not surprised that these cases come out in the community, but we always keep an eye on them to ensure that there's no outbreak leading to an epidemic following that. Like all sections, these are the section objectives to review following the lecture and to fill out an answer to make sure that we're getting the main points of these lectures. And that is the end of the lecture.